Joseph Minich. I'm the Director of Studies here at Freiburg Academy, and I'm so proud to introduce and award the prizes for tonight's first annual, possibly ever in the history of Freiburg Academy Science and Engineering Fair. So thank you again for all being here. Thank you for the participants. Before we get started, I just want to talk a little bit of cliche, because this is not an English contest like the speech contest. I'm allowed to use cliches. So I thought I'd start by defining science, and because that's the dorkiest thing I could possibly think of. So what is science? What does the word science mean? Of course, we think of science in all its facets today, right? Engineering, biology, human nature, behavioral science, environmental science, physics, relativity. Here's a little relativity joke. I am currently overseeing this event. You get it? Yeah? That's pretty good. Okay, good. Woo. I did not come up with that one entirely on my own. I'll tell you that. I'm a math guy. So, where was I? It's all relative. Okay, so science. The word science comes from a Latin word, right, that means to know, not surprisingly, and science is all about knowledge. But that word has deeper roots, much like plants, that we study in environmental science, deeper roots in an older Proto-Indo-European word, sorry for the English etymology lesson here, meaning to cut or to separate. And I think that's a perfect encapsulation of what the science fair has come to represent at our school this year. Right? We're separating knowledge, we're cleaving knowledge, right, into different categories, right? Both in terms of what the science has learned, what category of science it falls into, but also the quality of that science research. And so I think that's an instructive piece of information there to have moving forward, is that these are the best of our best, right? These are the best young scientists our school has to offer. You'll hear from a variety of different people tonight who've entered in state and regional competitions. You'll hear from people who've just for the first time ever involved in a science fair because our school has one and they were required to do so, but here they are successful nonetheless. So I really appreciate all the time and effort. I wanted to give a big round of applause to 3 people as well as some others, but first, I wanted to thank Dylan Harry, our science department chair, who's been instrumental in bringing this to fruition. There were times I wasn't sure we could pull it off, but his hard work, along with his compatriots Matt Fernier and Laura Meldrum, have been incredibly diligent and exhaustive and incredible. Just incredibly incredible. I guess that's, again, not an English event, so I'm fine on that. So thank you, Dylan. I'd also like to thank all the science teachers whose students contributed work to this event. Again, this is really a team effort, and it's been amazing to see the science department come together in such a cohesive way. Thank you to our judges and other supporters. We couldn't have done it without the judges, of course, and they've been deliberating for quite some time. They're finally done, and we're ready to move on to the awards. So before we do get to those, well, somebody's excited. Before we do go into those awards, we are going to have some project presentations from our main state science fair category awardees. So there are 3 individual groups, or 3 presentations by 5 individual students. So first up, we're going to have Jihu Lee and Jisoo Lee presenting on the effects of biochar on the plant growth and soil properties under heavy metal contaminated soil. So Jisoo and Jihu, come on up.
Hello, this is Jihu Lee.
I'm Jisoo Lee.
We are so honored to introduce our science project for this year to you tonight. And before we start, we just want to say thank you to Mr. Harry, Mr. Frenier, and Ms. Meldrum, who gave huge support to this project. So for this year's research project, we tried to integrate both of our interests, environmental science and computer science, by examining the effects of biochar on plant growth and soil characteristics in heavy metal contaminated environments. The idea was sparked when I came across a news report highlighting a significant decline in crop yields due to heavy metal contamination. This led me to consider the long-term consequences for farmers who are exposed to such adverse soil conditions, Motivated by this, we began searching for a solution that could help mitigate these effects and support sustainable agriculture. Then we found a substance called biochar, a soil amendment with the potential to improve crop resilience under these conditions. So the two main concepts used in this research are heavy metals and biochar. Heavy metals, intensified by human activities and climate change, are toxic contaminants that harm ecosystems and reduce agricultural productivity. They cause oxidative stress and inhibit photosynthesis in crops, with metals like copper significantly lowering yields. Agricultural soils are especially vulnerable to these effects. To resolve this problem, biochar has emerged as a promising solution, improving soil health by enhancing water retention, absorbing pollutants coming from heavy metals, and stimulating microbial activity through its remediation mechanism. Additionally, biochar reduces the bioavailability of heavy metals through its porous structure. Given its capacity to reduce the bioavailability and toxicity of heavy metals, this study aims to assess the effects of biochar on plant growth and soil characteristics under heavy metal stress, with a particular focus on its ability to stimulate microbial activity and its potential application is sustainable agriculture.
To investigate the potential of biochar in mitigating the adverse effect of heavy metal contamination, we designed an experiment focusing on plant growth under copper sulfate stress. Specifically, we selected biochar and copper sulfate as our primary variables, aiming to evaluate how biochar influences plant development in the presence of the common heavy metal. As shown in the diagram, we established two control groups with and without biochar, and four experimental groups exposed to two different concentrations of copper sulfate. Each group contained Bacillus burgers vinces, allowing us to systematically compare growth responses across treatments. Based on this setup, we conducted the experiment as follows. To begin, we applied the copper sulfate to the soil in the experimental groups. After planting the seeds, we observed plant growth over 20 days period, recording height once it reached over 5 centimeters. After the growth period, we examined root samples using the carbon fusion staining method, measuring soil pH values using soil to calcium chloride suspension method, and assess the root lengths. Using the collected data, we then constructed a predictive model to help people to predict the plant growth through the relationship between biochar treatment and plant resilience under heavy metal stress.
Among the various measurements we collected, those related to plant growth were particularly insightful in demonstrating positive effects of biochar. To evaluate how biochar affected plant development, we analyzed several indicators, including germination rates, plant height, growth rate, root length, and soil pH. Among these, three stood out, which are plant height, root length, and pH of the soil. As shown in the box plots, the median plant height in biochar-treated groups was consistently higher than in untreated groups, especially at higher biochar concentrations. Root length showed a similar clear distinction, with the most significant differences appearing under higher levels of copper sulfate, indicating biochar's protective effect under stress. Finally, the pH values of biochar-treated soils were noticeably higher in high copper concentrations, suggesting that biochar helped neutralize the acidity typically caused by copper cations.
To statistically assess the impact of biochar on plant growth, we tested the null hypothesis that there is no significant difference in plant height and root lengths between biochar-treated and untreated groups. Since both datasets failed the normality test using the Jerk-Vera test, we used the Mann-Whitney test, a non-parametric alternative to the t-test. While the height difference was visually noticeable, it was not statistically significant. However, the root length comparison yielded the p-value of 0.032, allowing us to reject another hypothesis and confirm a statistical difference in root lengths between two groups.
To further verify biochar's positive effects, we conduct root tissue analysis using the carbofusion staining method. Biochar-treated roots displayed vesicles, biofilm regions indicating microbial activity, and uniformly thin cortical cell walls. In contrast, roots without biochar showed irregular, thickened cell walls, suggesting structural damage caused by copper toxicity.
Finally, based on all the data we collected, we developed plant growth prediction model. We used Google Apps Script, which is a platform for developing websites using Google Tools, to create a web application that shows predicted plant growth depending on the presence or absence of biochar. When a copper concentration is entered into the input box, which is up there, the expected plant growth rate is automatically calculated using experimental data. As shown in the graph, we use the sigmoid curve to represent the plant growth. The red line represents the predicted growth rate with biochar, which is noticeably higher than the blue line, representing growth without biochar. Different input concentrations result in different predicted growth rate.
Taken together, our findings demonstrate that heavy metal contamination negatively affects plant growth by disrupting root-soil interactions and acidifying the soil, as shown through reduced plant height, shorter root length, and lower pH values of soil. Application of biochar effectively mitigate these adverse effects. Biochar-treated groups not only showed enhanced plant growth and healthier root structure, but also maintained more neutral soil pH levels, indicating improved soil conditions. These results suggest that biochar can serve as an optimal soil amendment to reduce the toxicity of heavy metals and support sustainable agriculture. Thank you.
Terrific. Thank you, Jisoo and Jihu. Next, we'll hear from Aisha Ribes on exploring plant microbial fuel cells for clean energy in Maine.
Exploring plant microbial fuel cells for clean energy in Maine was my science fair project that awarded me the first prize in the plant category in the Maine State Science Fair. The big question is that more than 770 million people on Earth live without electricity. But what if we could harness power from plant microbial symbiosis? What we will explore with my science fair project. Plant microbial fuel cells, also known as PMMCs, comparing C3 plants with C4 plants. C3 we use basil, and for C4 plant we use green onions. These two types of plants represent two different types of metabolism. We experimented their different treatments, including fertilization and artificial lighting. The experimental design. The conditions tested were fertilized versus not fertilized plant, soil-only controls, and artificial lighting from 7:30 a.m. to 5 p.m. in order to mimic winter's main climate. And the plant studies were basil, a C3 plant, and green onions, a C4 plant. In order to gather my data, I need to design, build, and program an Arduino setup because the sensors that I needed were more than $1,000 and I couldn't afford it. So I spent 2 months doing this design, in which I learned a lot about Arduino, some knowledge that will be useful in my future. The Arduino setup measures the voltage, the current, and the power every hour of each plant. For this, the Arduino make the measurement 10 times, average the value, and then introduce it into a data table. After 22 days, I had more than 12,000 data entries that I then analyzed statistically. The ANOVA analysis highlights that voltage and power differ significantly across different plants and treatments. Fertilization increased the energy output across both types of plant and also in the controls. And the control with fertilizer, so the highest energy production, something that at first didn't expect. Here we have the first graph, which represents the average energy production across different hours of the day. In the x-axis, we can observe the hours of the day, and in the y-axis, the power output. As we can observe, the plants had a diurnal cycle in which experienced much more energy production during the hours that the lights were turned off, and after these lights were turned off, the energy output declined rapidly. The dark blue represents the control with fertilizer, and the light blue the green onions with fertilizer. Then we have this second graph, which is a 95% confident graph, which shows the mean values of both power and voltage across the different types of plants and treatments. And the black line represents 2 standard deviations. This standard deviation seems to be really large, but this is due to the diurnal cycle that the plants experience, producing much more electricity during the daylight than during the night. And what are my conclusions? C4 plants produce much more electricity than C3 plants. This is due to the metabolism of these plants. C4 plants, as green onions, are more photosynthetic, efficient. They have a more efficient water use and ion use. This leads to higher moisture in the soil and quantity of ions, which leads to a greater conductivity rate and therefore energy production. We also observe that fertilizer boosts the microbial activity, leading to more power output. On the other hand, we have the challenges of C3 plants or basil. They have higher water loss during the day, and they recover this moisture more slowly during the night, leading to lower moisture levels in the soil and therefore conductivity rates. Because there is less ion movement, they also uptake more nutrients and ions from the soil, also decreasing the conductivity rates. And C3 plants have also another challenge, which is that their energy production drops quickly after the lights are turned off. Then we have the role of fertilizer, which brings agents and voltage across all types of plants, but it increases much more the electricity in green onions than in basil. In basil, it does not have as much effect. This effect is due to the ions that are contained in the fertilizer, since they are composed of nitrate, phosphate, and potassium salts that dissolve in the water. Then we have the effects of artificial light, in which boosted the energy across all types of treatments, making the power increase steadily during the day and peak from 10 a.m. to 3 p.m. The green onion sustained energy for longer after the lights were turned off. However, basil have a sharp drop after 5 p.m. when the lights are turned off. This leads to a higher power output in green onions. And then we have the control group mystery, which produced the most electricity overall, winning to the ones that had plants. And you might ask, why? The first reason is that there is no nutrient uptake, so there are more ions available in the soil, leading to higher conductivity rates. The moisture also keeps more stable, since there is no plant that take water content from the soil, and there are not as many biological cycles affecting the energy output. However, we hypothesize that in the long term, the control group will have an energy production decrease, since there will be no symbiosis between a plant and the microbes, so the microbes will lead to a nutrient depletion, since there is no plant that reintroduces carbons and ions into the soil. So my conclusions are that green onions produce more energy. Fertilizers increase energy output across all groups. Soil conductivity was a major factor. Green onions are the best energy producers. Basil loses water faster, leading to lower conductivity rates. Fertilizers boost output, especially in C4 plants, and control soil has high short-term energy output, but we hypothesize that in the long term will have less energy output than the other treatments. For future research, we should test more plant types and species, study how microbial activities influence the energy production in these PMMCs, improve long-term PMMCs energy efficiency, and study the long-term behavior of the controls, since we hypothesize that they will decline their energy production. Here we have some photographs of the process: building the Arduino, watering the plant, gathering them, and programming the Arduino. And here we have a video.
The Arduino setup with the plants here, and now it will start measuring.
And here we can
It
see.
measures every hour. That's plant 1. And you can see it goes to measuring each plant. It does that with the 10 plants we have, including the controls. So this is the Arduino setup.
Thank you. If you have any question, don't doubt in asking them.
Thank you, Isa. And last, we'll hear from Simon Yo and Nung Wing on the sustainable absorbent materials from sugarcane bagasse and chitosan for methylene blue removal.
Hi everyone, my name is Zong.
And my name is Simon. Today we will present to you guys the project we bring to MSSF this year, which Mr. Minich already read. Sustainable absorbent material from sugarcane bagasse and chitosan for methylene blue removal.
So, as we all know, water is a very important life source for all living things, including us human. And water pollution is getting worse and worse. One factor leading to that problem is factories are dumping out wastewater into the environment, and methylene blue is one of the substances inside it. So, what is methylene blue? Why is it so harmful? Well, methylene blue is widely used in textile industry and in medical use too. However, when a human intake methylene blue, it can only be considered safe when used at a small dose. With a larger dose, it can cause symptoms like nausea, dizziness, vomiting, and even cancer, which is very bad. In Vietnam, for example, factories are dumping out 8.2 times the standard limit set out by the government. They're dumping out methylene blue. Thus, me and Simon are trying to develop a sustainable and efficient way to this problem.
Yeah, that's right. That's what we do. There was a lot of study were executed and a lot of material that have been explored for the methylene blue removal. However, the product come out of these methods are often small material and hard to recover. Therefore, have little industrial applications. During the summer, there was one paper that caught me and Zoom eyes, was the paper that talked about how cellulose modified material show potential to remove harmful substance out of waters. That's why we decided to go into this direction. And the further we study sugarcane, readily agricultural waste, with its high cellulose content, we believe it is the worthy candidate and the best fit for our project mission, which we aim to synthesize a composite material that derives from, of course, sugarcane bagasse, chitosan, and sodium aminate. We would assess its effectiveness in removing methylene blue and also how different factors affect its characteristic.
So, fortunately, most of the material are very easy to find in our school, thanks to our school lab, except for the sugarcane bagasse, which is not available in Maine. We have to ship it all the way from South America to here.
Yeah. Sorry. So you guys see a couple of slides ahead, but yeah, we will go over these 4 steps right here where we synthesize our material.
So first, the bagasse collection. We collect our bagasse by, so first of all, we chop off the sugarcane into small pieces, and then we drain all of the water out to make the bagasse. After that, we wash it with a solution containing 50% ethanol to remove any dirt inside the bagasse. And then we dry it at 60 degrees in the oven for 24 hours to completely dry it. Sequentially, we treat the bagasse with a solution of NaOH and H2SO4 under reflux at 100 degrees Celsius for 1 hour to remove any lignin and bleach the bagasse. The purpose of that is to basically expand the surface area of our material. Next off, we wash the product with distilled water, and then we treat it again with a solution of H2O2 and NaOH to further remove any lignin inside our product.
So after the step that Zoom just said, we now have the cellulose. Then we come to the fun part, which we ready to synthesize our material by coating it with chitosan and sodium iodinate. So first we take the cellulose, put it in the chitosan solution, stir it at 1000 RPM until the mixture becomes homogeneous. We then take the mixture, put it in the syringe, and dropwise it in 0.5 molar NaOH solution for the material start to form. After that, we filter the material out, soak it further in 2% sodium iodinate for 2 hours. And then we have our material, as you can see in the picture right there, and we are ready to use. Fortunately, we were able to synthesize more than 50 grams of material from just 8.3 grams of the cellulose, which is really, really impressive. When examining the material structures under microscope at 40x magnification, it seems like our material going in a really good direction. You can see at these two pictures right here, which show a lot of changes, rough and porous surface. What it does is allow our material to have a favorable condition for methylene blue to be attached to it. Come back to the question of how well the material works and what is the best condition for it to work in. We then initially create those 9 bottles right there, which are the combination of 3 different pH values and 3 different concentrations. So the pH value is 2, 7.3, 10, and 5, 10, and 25 parts per million for the concentration. From those bottles, we insert 30 milliliter sample out and testing it at different time scale of 1, 6, 12, and 24 hours. With each 30 milliliter sample, we are using 0.1 gram of our material to test it out and collecting our data by using spectrometers as the wavelength of 625 nanometers. Here are some of the pictures after we soak the material in, retreat it, and it's down in the surface area again. And the figure 9 right here, as you guys can see, there's the color difference between the original bottle and after we treat it with our material. So the material is working. But as a scientist, we need to look at the data, right? And we have like more than 30 graphs, I believe, because there's a lot of combination. And it would be boring if we talk again about all of it tonight. And you guys probably won't hear about that. So I just gonna choose some notable graphs to talk to you guys tonight. First, let's look at how the concentration affects our absorption efficiency. When the concentration increases from 5 to 25 parts per million, as you can see right here, the blue ball, which is the efficiency, is decreasing, while the red line, represents for the capacity, is increasing. Why is that? You see, because in the lower concentration, there's less dye content, and the absorption efficiency of the dilute solution is, of course, always higher than that of the high concentration solutions. So another way to say it is, at the lower concentration, we absorb more percentage of it. Why? At the higher concentration, we absorb more amount of it. So that is different. But now you guys might wonder, why the efficiency is so low? 20%? You guys don't want to buy a product that have like, 20% efficiency, right? It's because we are using the 2 sample as the pH of 2, as you guys can see on the title. And let's dive deeper and see. Ooh, too far. And dive deeper and see how pH value affect our absorbent efficiency. So you can clearly see there's a pattern right here. When methylene blue dissociate into water, it will create methylene blue plus. And when the pH value is greater than 7, that means the negatively charged surface of our material is the favorite place for methylene blue to visit and attach to. That means in the lower concentration, lower pH value like 2, there's more competition between H plus and methylene blue plus to be attached to our material. And at the pH of 10, we even reach 100% efficiency. Really, really, really high. And I will hand it off to Zoom to talk about the last factors.
Thank you, Simon. So we're going to talk about how time affects efficiency and the capacity of our product. So imagine our product is like a ball with a lot of like active site on it. When methylene blue attached to the ball, which means like attached to the active site on the ball, there will be less active site. So that explains why from 1 hour to 6 hours, our methylene blue intake of our product, it's really high. You can see the slope is very steep. So this is because like initially our products have a lot of active site on it, so methylene blue will trying to attach to all those active sites. But after 6 hours onwards, the rate will be slowed down. Even though it doesn't really look increasing, it's still increasing. This is, by the way, because like the active site, most of them have been taken already, so methylene blue would have to find their way into a new active site to be attached.
Yeah, like worth to mention that our product is also time dependent. So as you can see, when the time is, the more we put the product in from 1, 6, 12 to 24, the blue bar is increasing. This is because, the product is time dependent, but like Zoom said, after 6 hours, the capacity reach its maximum, like increasing, but at a really, really slow rate. And therefore, after 6 hours, the more we put the material in, is like not effective and seems a little bit redundant. And is all of the factors we want to talk about, and here's the little table to capture what we just said. With the amount of data we have, we was able to also play around with kinetic model, which developed by other scientists to specifically testing the efficiency of the absorbent material. And after much calculation on Google Sheet, it seemed like the pseudo second order, which is the equation on your right hand right there, fit with our data. And what does that mean? Is that our material work by chemisorption. And chemisorption is that methylene blue will attach to our material through chemical interactions, and not just like simple diffusions like the first order. And this is good because it's stable and allows us to capture more methylene blue. The model also comes with a really, really steep slope. 0.2183, really low value. What does that mean is the reduction rate is like this, happen really fast. And explain to why Zoom say from 1 to 6 hours, the methylene blue,
the reaction is very high.
Uptake our material really, really, really fast. And as a result, as you guys can see, we did successfully synthesize a material which have rough surface, many grooves and pores, and the most important thing is it's not dissolved in water, so we are able to recover it. Zoom, you want to talk?
So surprisingly, our material work with an efficiency of almost 87%, at an optimal pH of 7.3, absorption time of 6 hours, and the concentration of the methylene blue is 5 part per million. Why do we choose pH of 7.3, even though you guys see that the pH of 10 have 100% efficiency? Well, we don't choose pH of 10 is because normally the wastewater are being dumped out would have the pH range of 6 to 9. So that's why we are choosing a pH of 7.3, because that is domestic water pH. Also, our material not only show that it can absorb methylene blue well, but it also shows that the potential to absorb other contaminated substance inside water, like heavy metals such as lead or iron, due to its
negatively charged surface.
Negatively charged surface. Simon, do you want to talk about future research?
Of course. So we actually have a lot of plans for future research because we cannot do everything during this project. So in the future, of course, we want to further explore our structures by using modern equipment such as FTIR, TGA, SEM, or XPX. We also want to optimizing our material synthesis process and use more of the more precision data measurement. So right now we are using the spectrometers with the maximum wavelength as secant rate 25. But when we are doing research, our aim is to capture the data as secant rate 65 nanometers. So with the better data, we will have a chance to play around more with more complex kinetic model. And if we have a chance to repeat the project again, we would absolutely test at larger scale, repeating the absorption experiment with intense precision and with more variability. And by this, we are closer to our goal of industrial application, because that is our main goal. And that is our project. And we would like to thank Mr. Harry for being a great PI this year. Thank you, Mr. Premier, Mr. Drew, for the support. And we would like to thank Mr. Manning, Mr. Fast, Mr. Stronger, Mr. Grant, Alvin, Matthew, Lawrence, for the valuable discussion and helpful comment. This is my last year with me and Zoom in this school, so we hope after today, after this science fair, I hope that the future generation of private academy will achieve even more successful result in the futures. And I would like to thank my roommates as well. Without him, I cannot do anything. So, yeah, thank you guys.
Thank you both for that excellent presentation on your project. Okay, we're going to move on to the awards. So we have a number of different categories of awards, as well as different categories in which these awards were awarded. My helpers are going to, my lovely assistants are going to come out, I think. Lovely assistants. Okay, first up, First place in the health sciences category goes to Helen Wu Che and Vivian Liu Che for the role of metformin controlling glucose levels. It doesn't bother me. Congratulations. First place in the biological sciences category goes to Elise Dunn for analyzing E. coli survival under rising river temperatures. First place in the physical sciences category goes to Tomono Arai for the effect of transporter mutation of potassium on radiocesium distribution in Arabidopsis thaliana based on the Fukushima nuclear explosion accident. Congratulations, Tomono. First place in the plant sciences category goes to Matthew Kim for effect of hydropriming on A. amylase or alpha amylase activity and germination for red kidney beans. First place in the behavioral and animal sciences category goes to Joseph Delavala for Nomophobia Nation and Phones Outperform Focus. First place in the engineering category goes to Aitan Solimez for detecting sign language using machine learning methods and AI. Congratulations, Aitan. And first place in the environmental science category goes to Alvin Darby. Analysis of polyhydroxyalkylamine production with the nutrient deprivation of spirulina potensis and porphyridium cruentum under both separated and co-culture conditions, the longest title and the winner. And then I'm going to invite up our MESA students. So for those of you who aren't aware, MESA is the Maine Environmental Science Academy. It's a program at our middle school here in town that has been a long-running program that allows students to investigate science, environmental science in particular, in conjunction with other academic disciplines. So these students have chosen their own awards for tonight's participants of the science fair. So they're going to come up and read those names. I'm not sure who wants to read them. Alex, I think you got elected, buddy. All right, so I'm going to give the stage right to them. Come on. Just stop right there. You can bend it down here.
Yep. So for third place for the MESA Student Peer Choice Awards, we would like to award Anna Carbone.
And in second place, we have
Joseph, how does it? Delavala. Joseph Delavala.
we would like to award first place to John Delavala.
It would not surprise me if all of those students were standing up here receiving awards in the near future. All right, we have 2 more categories of awards. The first of those is the Emerging Scientist Grants. So I'm going to have Ms. Meldrum and Mr. Frenier come up to the stage to present those. Is that what you want to do? Sure, why not? Come on up, you can hand them off. So these awards are for new science fair competitors who show promise and enthusiasm. As Ms. Meldrum commented, it's easy to teach science when there's enthusiasm already present. And this award includes a $200 grant for each of these students from the Freiburg Academy Science Department toward next year's science research project. Ready? Okay. So the 3 emerging scientist grants this year go to An Wing, Win Liu, and Lily Coriarty. Congratulations to all of you. We look forward to seeing your projects next year. And before we get to these final awards, we want to give a shout out to Larry Park and Harry Lee, whose projects using chitosan hydrogels for the removal of chemical pollutants from aqueous solution just won the Best in Maine for the Stockholm Junior Water Prize and has earned them a trip to compete in nationals in St. Louis this summer.
To hold them on stage. 1, 2, 3.
Oh, absolutely. Okay, so before we announce the finalists, just a couple instructions for those final 3 prize award winners. Just come up on stage and stand over here so we can get some really nice pictures of everybody with Mr. Harry and whoever else wants to join Mr. Harry. Okay. All right, so these awards do come with a wonderful cash prize that mimics our prize speaking competition. So the final awards for 3rd prize is $50. For second place is $100, and for first place is $150. And that can be used for not just scientific research, that's just straight cash. So 3rd place overall goes to Joseph Delavala. Make sure the right one. Yeah, over here, Joseph. There, next to Mr. Harry. Oh, we can take the thing. There you go, bud. Second place overall goes to Tamona Arai. And first prize in our first and certainly not last Freiburg Academy Science and Engineering Fair goes to Aitan Soilmez. All right, so that concludes this evening's competition and presentations. Do we get a good picture? Somebody get a picture?
I guess we're getting. Got a picture?
Okay. Somebody got a picture back there? Okay, good.
Just ask students to break down their posters.
Sure thing. All right, we ask our student participants to please break down your posters after the show and put them in their tubes, which sounds like an experiment unto itself. Thank you all for coming tonight. It's been a wonderful experience for, I think, all of us at the Academy to see the level of scientific research and interest and enthusiasm that our students can present when given the proper format. And so thanks again for coming out. And if you have any Ideas for next year, start thinking about them now. All right, have a great night.